Welcome to Matri Academy. Let's begin the chapter 26 Plant Genetic Engineering. The term biotechnology was coined by Early Erenki, a Hungarian engineer in 1999. The era of biotechnology started in 1973 when Herbert Boyer and Stan Cohen developed the rDNA molecule. The capacity to introduce and express foreign genes in plants was first described in tobacco by agrobacterium mediated gene transfer technique by Horst et al. 1984, D. Block et al. 1984, and by vector-less approach by Suzuki et al. 1984. The first genetically modified crop was tobacco with virus resistance gene in 1984. The cat gene was the first bacterial gene to be introduced into plant cell by Harry Estrella et al. In 1983. Transgenics. What is transgenics? Plants having genes incorporated from some other organisms are called transgenic plants. Cisgenic. Plants or organisms having additional gene incorporated from the same organisms. Cauliflower mosaic virus genome size is 8024 base pair. Gene 2 and 7 can be manipulated. Question asked in IRI 2003. Gemini virus has a genome size of 2.6 to 3 kilobase pair. Its core protein gene can be manipulated. Gemini viruses are having single-stranded DNA genome, while colivirus have double-stranded DNA. Question asked in ICR JRF 99 and IRI 2000. Virus bacteriophage 174 is having a single-stranded DNA and its genome size is 5373 base pair. Question asked in IRI 2003. Cytoplasmic male sterility is due to the mitochondrial genome. Cytoplasmic male sterility or is used in hybrid seed production. The 35S promoter is the most commonly used constitutive promoter both in monocots and dicots. Transfer of naked DNA is known as transformation. Question asked in ICR JRF 1999. Linearized DNA is much more effective in producing stable integration than circular DNA. Electroporation is the process where electric impulses of high field strength are used to reversibly permeabilize cell membranes to facilitate the uptake of large molecules including DNA. In electroporation, a doubling of the chloroplast diameter would reduce the optimal voltage to 1/2. Electroporation aids in transformation by forming small transient pores in cell membrane for the easy uptake of foreign DNA. Question asked in ARS 2007. The particle bombardment is the most preferred method for monocots. Serials transformation, also the best for plastid or chloroplast transformation. Question asked in ARS 2012. Particle bombardment technique is the most versatile and effective way for development of transgenic plants. The microprojectiles used are variety of metals such as gold, tungsten, palladium, rhodium, and iridium of 1 to 3 micrometer diameter. The first successful chloroplast transformation in higher plants was achieved in 1989 when spectinomycin resistance gene was transferred into tobacco. During liposome-mediated gene transfer, it involves first the collision of DNA molecule with the protoplast followed by endocytosis of the DNA molecule by the protoplast. Question asked in DRS NET 2001. PEG, polyethylene glycol, and calcium phosphate are thought to precipitate the DNA onto the outer surface of plasma lemma, and the precipitate is taken up by endocytosis. Which chemical is used to make the bacterial cells competent during genetic transformation experiment? And the answer is calcium chloride. Question asked in ARS NET 2001. The most commonly used method for transformation of plant is agrobacterium tumefaciens mediated genetic transformation, while for animals, Transportation microinjection method is the most preferred one. This is important. Agrobacterium tumefaciens induce crown gall formation. Question asked in TRS 2007. TI plasmids are present in Agrobacterium tumefaciens. Crown gall by Agrobacterium tumefaciens is caused because of phytohormones TMS1, 2, TMR and IPT gene. Question asked in IRI 2003. The region of active Agrobacterium tumefaciens responsible for tumorigenesis is tDNA. Question asked in IRI 2003, CSIR June 2004, and it is of 25 kilobase pair. The size of TI plasmids is 200 kilobase pair. Molecular weight is 1.2 into 10 to the power 8. VIR region weight 40 kilobase pair having 24 genes arranged in 8 operons from Viron A to Viron H. The size of tDNA is 23 kilobase pair in Neapolitan type TI plasmid, and in octapine type TI plasmid it has two adjoining regions of 30 kilobase pair left hand piece and 8 kilobase pair right hand piece. The size of TI plasmid is 200 kb, tDNA का 23 kb, PRI plasmid का 250 kb and right and left base का 24 base pair each. Question asked in CSIR June 2003 and NET 2014. tDNA can accept a DNA insert of greater than 50 kilobase pair size. In tDNA, left and right has got 24 base pair size. Question asked in TRS 2007. The 25 base pair flanked region of tDNA is stimulated by Viron D region of TI plasmid. Question asked in TRS NET 2001. The tDNA most likely enters the nucleus through nuclear pore complex. Question asked in TRS 2004. tDNA integration take place by random recombination. Question asked in IRI 2003. When Agrobacterium infects a plant cell, then the plant cell starts synthesizing an arginine derivative known as opines. ARS NET 2001. Octopine synthase precursor is arginine, pyruvic acid and NADH, and nopalin synthase precursor is arginine, ketoglutaric acid and NADH. Question asked in TRS 2018. During Agrobacterium mediated genetic transformation, antibiotic cefotaxime is used for suppressing the growth of agrobacterial cells. By killing them, an antibiotic kanamycin is used as selection agent to select the transformed cells from the non-transformed ones. Question asked in TRS NET 2001. Acetosyringone is released by the cut end of X plant, which induces the expression of Viron aprons, especially in dicot plants. Where is monocots, we have to add acetosyringone into the medium for Viron operon induction and tDNA transfer into the plant cells, since monocots do not produce acetosyringone. Question asked in TRS NET 2001. For tissue-specific expression of gene in plants, we use tissue-specific promoters. Example, SF8 promoter for expression in seeds in sunflower, while for expression of genes throughout the plant system, we use constitutive promoter. Example, cauliflower mosaic virus 35 as promoter. Question asked in TRS NET 2001. The gene AAD, O-aminoglycoside adenyl transferase, allow the selection of bacteria containing it on a medium supplemented with spectinomycin or streptomycin. NP2 gene is of limited value in monocots due to their tolerance to relatively high level of kanamycin. This may be the reason of lack of transformation work in monocots. Using NP2 gene in Agrobacterium species as selection marker. Origin of Gus gene in UIDA locus in E. coli. IRI 2003 NET 2013 question. The sequence of lac operon is ZYA. IRI 2003 question. Luciferase produce distinct phenotype but do not allow selection. GFP, gluing fluorescent protein gene, is a scorable marker gene and not an antibiotic resistance gene. ARS NET 2001. Race specific or pathotypic specific resistance is called vertical resistance. Tomato leaf curl is caused by virus. IRI 2001. Plant Gene Bank in India is located at NBPGR New Delhi. ICRGRF 2004. Negative selection. In this process, the untransformed tissue die. Example by selection pressure posed by kanamycin. Positive selection. In this process, the non-transformed tissue do not die, but they do not grow or proliferate. Example selection posed by phosphomannose isomerase gene. Phosphomannose isomerase is used as a positive selection agent. Question asked in ARS 2007. For confirming transgene copying number in any transgenic plant, we can use real-time PCR, also known as quantitative PCR or qPCR, in addition to southern hybridization. Clean gene technology. It is a marker gene-free technology. No selectable marker will be used during genetic transformation, or if used, then they will be excised later on using various techniques like Cree-Lock system, co-segregation, etc. Eco-friendly technology had poses no environmental threats. For the characterization of transgenic plant or detection of homozygous and heterozygous plant, we use event PCR. In this variation of PCR, we design one primer from the transgene construct and the other from the flanking genome sequence, also by tail PCR and fish PCR. Immunolocalization technique can be used to locate the expression of a transgene in a particular cell at a particular stage of development. Bacillus thuringiensis. Bacillus thuringiensis was isolated first from the disease larva of silkworm Bombyx mori in Japan by Ishiwata in 1901. It was not described officially. The pathogen Bacillus thuringiensis species Kursatakai was probably the first described from diseased silkworm by Pasteur sometimes in 1960. However, it was re-isolated by Berliner in 1915 from diseased larva of Mediterranean floor moth Angasta species in Thuringia in Germany. Hence the deviation of species name Thuringiensis. 60 SSP of Bacillus thuringiensis have been classified, containing over 140 described crystalline toxins. Bacillus thuringiensis species Kurastaki, Ajwai, Marosini, and Israelensis. Bacillus thuringiensis also produce delta enterotoxin, an enzyme which enhances the activity of endotoxin. Bollworm is a serious pest of cotton. ICR-JRF 2004. Cry1AC gene imparts resistance against lepidopteran insects. ARS 2007. Most of the cry genes are situated on large conjugative plasmids. Bacillus thuringiensis produce insecticidal cry proteins. IRI 2003. So far, the only commercialized transgenic crop in India is BT cotton. ARS 2007. First commercialized transgenic crop in India was BT cotton containing Cry1AC gene, released in 2002. IRI 2004, 2011, ARS 2004 question. For developing BT cotton hybrid, Coker system was used. BT cotton Bolgard 1 consists of Cry1AC gene. ARS 2007. Bolgard 1 cotton contains Cry1AC gene for imparting resistance against cotton bollworm, while Bolgard 2 contains two genes Cry1AC, Cry2AB for imparting resistance against both cotton bollworm and pink bollworm. The cotton bollworm Helicobra almigara and pink bollworm Pactinophora glycophyllum. Question asked in IRI 2011, ARS 2007 and 13. ईल गार्ड बीटी मेज में क्राई 1AB जीन प्रेजेंट होता है एंड स्टारलिंक बीटी मेज में क्राई 9C जीन्स आर प्रेजेंट. क्वेश्चन आस्क्ड इन ARS 2003. Heliothis armigera is a polyphagus pest. Lathyrus is a pepsin inhibitor. ICR-JRF 2001. The baculovirus contains mostly DNA as a genetic material. ARS-NET 2001. Most of the baculovirus belongs to the order Lepidoptera. ARS-NET 2001. The baculoviruses are used as biopesticides because they have a small residual life as compared to chemical pesticides. ARS-NET 2001. The gene chitinase can be effectively used for imparting resistance against fungi. ARSNet 2001. Defense response in plants can be induced by acetyl salicylic acid. ARS 2007 question. To develop virus resistance, squash melon coat protein gene was used. ARS 2007. The transgenic plants having incorporated virus resistance for tropical countries was papaya. ARS 2007 question. The techniques in which one strand of DNA, that is sense strand, is made antisense and the antisense strand is made sense and thus we confer resistance against viruses is known as antisense RNA technology. Question asked in ARS NET 2001. The most commonly used techniques for imparting resistance in plant against viruses is antisense RNA technology. ARS NET 2001. The principal compound produced by host cell in response to the virus infection in plants is an antiviral protein known as interferon. Question asked in IRI 2000 and 2004. Ribozyme is an RNA molecule which has enzymatic activity usually concerned with DNA degradation. Example is satellite RNA of tobacco ring spot virus. Question asked in IRI 2004. Most of the animal viruses contain double stranded DNA and most of the plant viruses contain a single stranded RNA. Flavor sour tomato had been developed by antisense suppression of polygalactourinase gene by Calgene USA in 1995. ARS NET 2001 and 2007. It was the first commercialized genetically modified crop in the world, sold by the name of Magriger. ARS NET 2001, ARS 2012 question. So note it. The most successful example of modification of oil quality is the increased lauric acid content of Brassica napus. Lauricol is the first commercial variety of Brassica napus having modified oil composition by engineering of the gene lauryl ACP thioesterase. RNAi technique for the first time was reported in Caenorhabditis elegans and nematode in 2006. Question asked in ARS 2007. miRNA produced from RNA molecule with imperfect stem loop structure. siRNA produced from long double stranded RNA molecule. It is 21 to 23 nucleotides long. Golden rice developed by Indopotraches for increased vitamin A content. Rice variety was Tapi 309. ARS 2013 question. Father of golden rice technology is Ingo Potricus. Cereal seed grains are deficient in lysine, while those of pulses are deficient in sulfur-containing amino acid, cysteine, and methionine. IRI 2001 question. Development of transgenic plants resistance tolerant to herbicide was the first major achievement of genetic engineering in plants. Glyphosate inhibits chloroplast enzyme 5-enol pyruvyl shikimate 3-phosphate synthesis, that is EPSPS, encoded by ARO-A gene of chloroplast genome. Roundup Ready maize has been engineered for glyphosate resistance. ARS 2007 question. BAR is a herbicide resistant tolerant gene obtained from Streptomyces species and encodes for phosphophenothricin acetyl transferase, that is the PAT. Question asked in ARSNet 2001 and 2004. Herbicide atrazine and bromoxynil inhibits photosystem 1, while paraquat inhibits photosystem 2. Sulfonylurea inhibits the enzyme acetolactate synthase, and herbicide glyphosate inhibits the enzyme EPSPS. Herbicide resistance tolerant gene along with their source. BAR gene, Streptomyces hygroscopicus and Streptomyces viridochromogenes, and effective against Biolophus basta and glufosinate. ARO-A gene is present in E. coli or Salmonella typhimurium and effective against glyphosate. ALS mutant present in E. coli in yeast and effective against sulfonylurea and imidazole. the BXN gene Klebsiella osazone, and it is effective against bromoxynil. Lee et al. In 1955 produced high temperature, stress tolerant transgenic Arabidopsis plants by altering the level of expression of heat shock protein. By changing the expression level of Arabidopsis heat shock protein transcription factor ATHSF. Question asked in TRS 2001. DREB, dehydration responsive element sequence, a transcription factor. This protein causes expression of all the genes taking part in moisture stress. The oral cholera vaccine consists of recombinant CTB, chlorotoxin B subunit, and killed Vibrio cholerae cells. Question asked in CSIR December 2002. The minimum length of homology reported between two homologous sequences for inactivation is 90 base pair. Transfer of genes between different species is known as horizontal gene flow. Transfer of transgenes can be done from one plant to other cultivated varieties by backcross breeding. The authority for approval and release of transgenic crops for commercial purpose is the ministry with the GEAC, Genetic Engineering and Appraisal Committee of the Ministry of Environment, Forest and Climate Change under the Environment Protection Act 1986. Question asked in the ARA 2018. Which genetically modified crop has got the highest area in the world? That is the herbicide tolerant soybean. Carotene Protocol on Biosafety, an international agreement that seeks to protect biological diversity from the potential risks posed by modern biotechnology. So, students, these all are the short notes, or you say it is the quick notes about the genetic engineering topics. So, it definitely get you to help in your the upcoming examination of ARS net. So, best of luck for your exams. I hope that you got success in your examinations. Thank you. Always like and subscribe my channels and let's tune to my channel.